Hello students, welcome to EPG Pathshala. I am Dr. Zinat Iqbal from Department of Pharmaceutics, Faculty of Pharmacy, Jamia Hamdard. Today we would be deliberating upon the module Factors Affecting Mucoadhesion and Evaluation of Mucoadhesion from the paper Novel Drug Delivery System 1. At the end of this module, we should be able to reach the learning objectives. The first one, I think we have to understand this fact again, that mucoadhesion primarily is the premise for all the drug delivery systems which needs to be adhered onto a mucosal membrane. Then there has to be certain factors which will affect the mucoadhesion. Let us try to understand each one of them one by one. We will be talking about the factors which are polymer related, we will be talking about the environmental related factors and also the physiological factors which will affect the power of mucoadhesion. Later on, towards the end of the module, we would be talking about the various techniques of evaluation of mucoadhesions, namely the quantitative methods, the qualitative methods and the in vivo methods. Beginning from the first category or the first set of factors which will affect the mucoadhesion, as we understand, polymer makes the basis of the bioadhesive or mucoadhesive systems. The factors which primarily are because of the polymers include number one, the hydrophilicity, molecular weight, cross-linking and swelling, spatial conformation, concentration of the active polymer and the hydrogen bonding capacity. Then the other factors which will primarily have a major effect on mucoadhesion are headed as environment related factors. What will these include? These will include the pH, the applied strength. If we talk of the applied strength, the power or the strength by which a mucoadhesive drug delivery system is attached primarily to the mucous membrane. Then of course the moistening will matter, and primarily certain presence of the metal ions would also be an important factor. Then we come to the most important factors which will affect the mucoadhesion. We already understand that it is a biological membrane along with the dosage form which has the power of mucoadhesiveness. What are the physiological factors which will probably have a direct effect on mucoadhesion? They are supposedly the mucin turnover, the disease states, the rate of renewal of the mucoadhesive cells, the mucosal viscosity and perhaps the tissue movement. In the last three slides, we had just visualized the various factors which affect mucoadhesion. Amongst them, the first category is the factors which are polymer related. The first one or the first factor which is polymer related is hydrophilicity. Mucoadhesive polymers having hydrophilic functional groups such as carboxyl and hydroxyl interact with the mucosal surface via hydrogen bonding. On contact with the aqueous media, the polymer swells, resulting in exposure of potential anchor sites. Further, the swollen polymers have got the maximum distance between their chains, which lead to greater levels of flexibility and penetration of the polymer in the mucosa. The second factor, which is directly having the effect of the polymer choice, is its molecular weight. The mucoadhesive ability of the polymer depends on its molecular weight. The bioadhesive or mucoadhesive force of the polymer increases up to 1 lakh times with the molecular weight. However, no further enhancement is observed beyond this value. The next factor which we'll discuss is the cross-linking and the swelling capacity of the polymer. The density of cross-linking of the polymer is inversely related to its swelling. Low density leads to high degree of swelling and thus larger surface area and better mucoadhesion. It also leads to increased hydration rate and increased flexibility. However, increased hydration and swelling may lead to slippery surface of the polymer, which may result in its rapid removal from the biological substrate, decreasing the retention time. The mucoadhesion can further be increased by including adhesion promoters such as polymers grafted onto the preformed network and free polymer chains. The next one which we are discussing is the spatial conformation of the polymer. Spatial conformation of polymers determines its adhesive strength. Non-linear polymers or polymers with helical structures may shield many active groups responsible for adhesion. Example is dextran, which has a very high molecular weight, but the adhesive strength is very much similar to polyethylene glycol with a much lower molecular weight. This is because of the helical conformation of dextran as compared to the linear PEG. Concentration of active polymer. It has been found that the concentration of polymer influences its mucoadhesion. Beyond an optimum concentration of the polymer, a significant reduction in the mucoadhesive capacity is observed. In a concentrated solution, the coiled molecules become solvent deprived and the availability of chains to interpenetrate with the mucosal surface becomes less. This result is mainly applicable to liquid mucoadhesive formulations. However, for solid dosage forms such as tablets, higher polymer concentrations is usually resulting in stronger mucoadhesion. The next factor is the hydrogen bonding capacity. The hydrogen bonding capacity of the polymer is an important factor that determines its mucoadhesion or bioadhesion. The polymers must have functional groups that are able to form the hydrogen bond, which are usually responsible for having or eliciting the required bioadhesion. The next set of the factors which would influence the mucoadhesion of any drug delivery system are the environmental related factors. Amongst them, the primary one is the pH. The pH at the interface of the bioadhesive and the biological surface affects the bioadhesion or mucoadhesion, especially in case of the bioadhesive systems or polymers with ionizable groups. Several mucoadhesive polymers are polyanions possessing carboxylic acid functional groups which are used in drug delivery. If the pH of the microenvironment is more than the pKa of the polymer, it will be largely ionized, and if the pH is below the pKa of the polymer, it will be largely unionized. The maximum bioadhesive strength of polyacrylic acid polymer is observed around pH 4 to 5 and decreases gradually when it is above the pH of 6. There are numerous investigations which have shown that the carboxyl groups which are protonated rather than the ones which are ionized react with mucin molecules by forming hydrogen bonds with it. The next factor, which is an environmental driven factor, is the applied strength. For applying a mucoadhesive system, a definite strength is required. Higher shear force may lead to increased interaction, and higher bioadhesive strength, it affects the interaction of the adhesive with the mucus and hence determines the depth of penetration of the polymeric chains. Also, greater the contact time between the mucoadhesive and the mucosal surface, greater the swelling and greater is the interpenetration of the polymer chains. The next environmental factor which influences the performance of a mucoadhesive system is the moistening. Moistening or wetting is required to allow the mucoadhesive polymer to spread over the mucosal surface and create a macromolecular network of sufficient size for the interpenetration of the polymer and mucin molecules and to increase the mobility of the polymer chains. Therefore, it is important that proper moistening or wetting is there if we require a best performance of the mucoadhesive drug delivery system. The last factor, which primarily is also based on the environment, is the presence of the metal ions. Ionic strength of the surrounding environment affects the mucoadhesive bond. Interaction with charged groups of polymers and or mucus can shield the number of interaction sites required for adhesion and tightness of the mucoadhesive bonding. However, some polymers like Gellan are reliant on the presence of divalent cations for in situ gelation. Students, by now I think we are able to appreciate that the major effect of the factors which will be on the mucoadhesive drug delivery systems are of the category of physiological. The reason why they are supposedly the most terrific or the most challenging factors, because these are not under the control of the formulators. Let us try to understand each of the factors one by one. The first and the foremost factor is the mucin turnover. The mucus turnover rate varies in disease state or in the presence of food. Then it limits the residence time of the mucoadhesive system. More is the mucin turnover, more will be the slipperiness, and less will be the retention of the dosage form as such. Then the mucin turnover determines the contact time. This is what I just meant when I said that the retention will be affected. Then later on, it also results in increased amount of soluble mucin molecules which interacts with the mucoadhesives before interacting with the mucosal layer. The second physiological factor which will be of tremendous responsibility in the final performance of the mucoadhesive drug delivery system is the disease state of the individual. Properties of the mucus varies in various disease conditions such as common cold, gastric ulcers, ulcerative colitis, cystic fibrosis, bacterial and fungal infections of reproductive tract, and inflammatory conditions of the eye. Each of these disease conditions will definitely make a change in the mucosal membrane surface, and hence the ultimate performance of the dosage form would be definitely compromised. The mucoadhesives which are used in such conditions needs to be thoroughly evaluated. Then we have the next physiological factor, that is the renewal of the mucoadhesive cells. The rate of renewal of mucoadhesive cells varies from different types of mucosal surface and limits the residence time of the bioadhesive on the substrate. Then comes the mucosal viscosity. The mucosal layer viscosity varies throughout the body and also in the disease states. Low mucus viscosity results in weak interactions in the detachment and poor retention of the mucoadhesive drug delivery system. But highly viscous layer restricts the interpenetration and increases the diffusion path through which the active agent must pass. Then finally, we have another factor which is referred to as the tissue movement. The movement of tissues affects the retention of mucoadhesive system, especially in the case of gastroretentive systems. And in that case, when we talk of the gastroretentive systems, the primary tissue movement would be because of the peristalsis. It is now well understood that mucoadhesive drug delivery system primarily will only be successful if we have a choice of polymer which shows appropriate mucoadhesion. The performance of a mucoadhesive polymer would probably be tested by using different types of mucoadhesion techniques. These techniques have been traditional, which probably were used in the 1940s, and later on there was a progressive advancement and newer techniques were added in order to help the better estimation of the mucoadhesion power of a particular polymer. In the subsequent slides, we would be discussing each of these techniques and how they are applied for the evaluation of the mucoadhesion of a particular polymer. Methods of characterizing mucoadhesion. Several methods are available, both in vitro and in vivo, to screen and determine the mucoadhesive properties of the formulations. Park and Robinson, in the year 1984, were the first scientists to actually characterize the mucoadhesive properties of the putative polymers. They described the usage of fluorescent probes for estimating the mucoadhesion for a conjunctival epithelium. The latest development in the field suggests the use of force microscopy techniques like the atomic force microscopy, which are well suited to exhibit the molecular level mechanisms. Majority of researchers have used the basic technique of assessing the force required to detach the delivery system from the mucosal surface by disrupting the adhesive forces. The force so applied could be in different directions, thus leading to any of the three mechanisms of testing the bioadhesion force, namely tensile test, shear test, and peel test. Figure 1 depicts the different forces involved in the assessing mucoadhesion. The first figure shows the tensile force, the second one is the representative of the shear force, and the last one is talking about the peel forces. The methods of assessment of the mucoadhesions can actually be identified under the three major headings. We can classify them as the quantitative methods, qualitative methods, and the in vivo methods. We would now subsequently discuss the various quantitative methods. Amongst them, the very first one is referred to as the peel strength or the peel test. The test is based on the calculation of energy required to detach the dosage form from the substrate material, which is usually excised buccal mucosa attached to the bioadhesive material. The bioadhesion or mucoadhesion of tablets and transdermal patches is best found out using the peel test. The peel test offers a limited use in most mucoadhesive systems. Peel test is of value when the bioadhesive or mucoadhesive system is designed as a patch. In peel test, stress is focused at the edge of the joint, unlike in tensile and shear experiments in which stress is uniformly distributed over the adhesive joint. There are different types of peel test. They are designated as the T-peel test, the 90-degree peel test, and the 180-degree peel test. Amongst the one which is called as the T-peel test, it is the type of tensile test that is performed upon two flexible substrate which have been bonded together and are placed into a peel test grips such that one substrate sticks upside and the other sticks downwards, while the bonded area sticks out horizontally, so the entire system forms a T shape. The word T is usually because of the shape which is taken while the test is being performed. The next type of peel test is usually referred to as the 90-degree peel test. It requires a fixture of 90 degree to determine the adhesive strength between a rigid surface that could be a plate and a flexible that could be a tape, where the plate lies in a horizontal position with the gripped end of the tape sticking up perpendicular, while the rest of the system is bonded to the plate so that it forms the L shape. The last amongst the peel test is referred to as the 180-degree peel test. This is almost similar to the 90-degree peel test, just with an exception that the bonded area between the plate and the tape is placed vertically between the peel test grips, although the free end of the plate is held by the bottom and the free end of the tape is held by the top, so that it forms a firmly tight U shape. The diagram on the screen precisely emphasizes upon the shape which is taken while the substrate is being tested. The next mucoadhesion technique usually relies upon the premise of measuring the shear stress and the tensile stress of the formulations. It measures the force required to cause the bioadhesive to slide with respect to the mucus layer in a direction parallel to their plane of contact. Such forces act tangentially to a surface and can be calculated by dividing it by the area of the surface. It is the force per unit area required to sustain a constant rate of fluid movement. Smart and co-workers were the first group of scientists who had described the Wilhelmy plate method, primarily used for the study of the attachment of polymeric films to mucin. There are three typical definitions of tensile strength. They can be referred to as the yield strength, the ultimate strength, and the breaking strength. The yield strength is the stress a material can withstand without permanent deformation. The ultimate strength is usually referred to as the maximum stress a material can withstand, while the breaking strength is the stress coordinate on the stress-strain curve at the point of rupture. The next method is referred to as the colloidal gold staining method. It was first proposed by Park and Park in the year 1989. This method primarily employs the usage of red colloidal gold particles, which when adsorbed onto the mucin molecule forms mucin gold conjugates. Such conjugates, upon interaction with bioadhesive hydrogels, develop a red color on the surface. The intensity of the color is then quantified by measuring at 525 nanometers. The next method which can be employed is referred to as the direct staining method. It is primarily a novel technique which is used to evaluate polymer adhesion to human buccal cells following exposure to aqueous polymer dispersion both in vitro and in vivo. Adhering polymer is usually visualized by staining with 0.1% weight by volume of either Elchin blue or eosin solution, and the uncomplex dye is then removed by washing with 0.25 molar sucrose. The extent of polymer adhesion is quantified by measuring the relative staining intensity of control and polymer treated cells by image. Carbopol 974P, polycarbophyll, and chitosan are usually able to adhere to human buccal cells from 0.10% volume weight by weight aqueous dispersions of these polymers. Following in vivo administration as a mouthwash, these polymers persisted upon the human buccal mucosa for at least 1 hour. This method is only suitable for assessing the liquid dosage forms, which are widely employed to enhance oral hygiene and to treat local disease conditions of the mouth such as oral candidiasis and dental caries. One of the newest method is referred to as electromagnetic force transducer. This is EMFT. The EMFT uses a calibrated electromagnet to detach a magnetic loaded polymer drug delivery system from a tissue sample. It has the unique ability to record remotely and simultaneously the tensile force information as well as high magnification video images of bioadhesive interactions at near physiological conditions. The EMFT measures tissue adhesive forces by monitoring the magnetic force required to exactly oppose the bioadhesive force. The second set of methods which aim at measuring the mucoadhesion are under the category of qualitative methods. These methods are useful for preliminary screening of the respective polymer for its bio or mucoadhesion, compatibility, and stability. This tool was primarily used by Mikos et al. In the year 1991. However, these methods are not used in measuring the actual bioadhesive strength of the polymers. There are different set of methods which are employed under this, which include number 1, the viscometric method. Hagerstorm and Edwin et al. In the year 2003 had studied the dynamic rheological measurements on gels containing 4 different carbopole polymers and the corresponding mixtures with porcine gastric mucin and bovine submaxillary mucin. The method does not give the same ranking order when 2 different comparison strategies were used. The results were in contrast to the results obtained with the tensile strength measurements. The next method is referred to as the perfusion method and the rheological test method. Many researchers have developed different related methods based on the viscometric analysis. Amongst them, Hassan and Gallo developed a simple viscometric method to quantify mucin polymer bioadhesive bond strength. Their study concluded that the mixture of mucin and polymer exhibited much higher viscosity than the polymer or mucin alone. Later on, Murtazavi and Smart investigated the effect of carbopole 934P on rheological behavior of mucus and effect of various factors such as ionic concentration, polymer molecular weight, its concentration, and the introduction of anionic, cationic, and neutral polymers on mucoadhesive mucus interface. Carmela et al. Later on investigated the influence of polymer concentration and polymer mucin weight ratio on chitosan-mucin interaction assessed by means of viscosimetric measurements. It must be understood that the changes in the rheological properties of mucoadhesive polymer and the mucin may be due to some of the factors such as number 1, physical entanglement, that is the chain interlocking, which will ultimately lead to conformational changes. It could also be due to the chemical interactions, which includes non-covalent bonds, including such as hydrogen electrostatic hydrophobic bonding. Covalent bonds such as disulfide bridges could also be present. These occur alone or in combination between the 2 macromolecular species. The rheological evaluation gives information on the extent and magnitude of interaction between mucus and polymer. Since the increase in viscosity resulting from mixing of mucus and polymer has been claimed to correlate with mucoadhesion. This method per se should not be used as a stand-alone method for studying mucoadhesion properties of polymer gels. As discussed earlier, Hassan and Gallo had probably used this method to assess the viscosities of the combination of polymer and mucin. They proposed an empirical equation for the viscosity changes due to the interaction between the polymer and mucin, such as eta T is equal to eta M plus eta P plus eta B, where eta T is the viscosity of the solution containing both mucin and polymer, and eta M and eta P are the individual viscosities of mucin and polymer respectively, and eta B is the contribution that appears to be an outcome of molecular interaction between the polymer and mucin. It can be concluded that viscosity synergism could be used as a dependable in vitro parameter to measure and compare the mucoadhesion properties of various polymers. The next method in line is headed as analytical ultracentrifuge criteria for mucoadhesion. The scientist Harding SE in 2003 suggested the presence of various interactions within the polymer and mucin, primarily forming the complexes. The measuring criteria are based on finding the change in molecular mass using the sedimentation equilibrium. This marker has an accompanying limitation and is restricted to an upper limit of less than 50 mDa. Since complexes can be very large, a more sensible assay procedure is to use sedimentation velocity with change in sedimentation coefficient S as their marker for mucoadhesion. Where mucin is available in only minuscule amounts, a special procedure known as sedimentation fingerprinting can be used for assay of the effect on the mucoadhesives. UV absorption optics is used as the optical detection system. However, in this case, the mucoadhesive is invisible, but the pig gastric mucin at the concentrations normally employed is visible. The sedimentation ratio given by S complex divided by S mucin, the ratio of the sedimentation coefficient of any complex involving the mucin to that of pure mucin itself is used as the measure for mucoadhesion. The next method which we would discuss is referred to as the atomic force microscopy. This method is based on the changes in surface topography when the polymer binds to the buccal cell surfaces. Unbound cells show relatively smooth surface characteristics with many small crater-like pits and indentations spread over cell surfaces. While, on the other hand, the polymer-bound cells will lose crater and indentation characteristics and gain a higher surface roughness, which could be very well visualized by the technique of atomic force microscopy. The next method is referred to as the electrical conductance method. As the name suggests, it is dependent on the measurement of the electrical conductance. Sudhakar et al. In 2006 mentioned the use of modified rotational viscometer by Brimacher to determine electrical conductance of various semi-solid mucoadhesive ointments. He concluded that the electrical conductance was low in the presence of the adhesive material. The next technique which we will discuss is referred to as fluorescent probe method. The technique basically depends upon the use of the various types of fluorescent dyes. In the year 1984, Park and Robinson reported the use of pyrene and fluorescein isothiocyanate dyes to label the bilayered membrane lipid and membrane proteins. When mixed with mucoadhesive agents, the cell showed changed fluorescence spectra, which was monitored. These changes provided a direct indication of polymer binding and its influence on polymer adhesion. Lectin binding inhibition technique. This method involves an evident biotin complex and a colorimetric detection system to investigate the binding of bioadhesive polymers to buccal epithelial cells without having to alter their physicochemical properties by the addition of marker entities. The lectin concavalin A has been shown to specifically bind to sugar groups present on the surface of the buccal cells. If polymers bind to buccal cells, they will mask the surface glycoconjugates, thus reducing or inhibiting concavalin A binding. Thumb test is the next test which we will discuss. One of the simplest method, it relies on the measuring of the adhesiveness by assessing the difficulty of pulling the thumb from the adhesive as a function of the pressure and the contact time. Although the thumb test may not be conclusive, it provides useful information on peel strength of the polymer. Dear students, there's an important point which has to be made here, that the final success of any formulation is dependent on how it behaves inside the animal model or the human body. In order to get close to such techniques, we have to assess every dosage form in order to find out whether it behaves nicely or according to the desire in the in vivo or it does not. Some of the techniques which are very suitable for finding the mucoadhesion in the in vivo conditions are, the first one is we can assess the GI transit using the radio-opaque technique. It involves the use of radio-opaque markers, example barium sulfate, which could be encapsulated in a bioadhesive drug delivery system to determine the effects of bioadhesive polymers on the GI transit time. Sometimes the fecal collection using an automated feces collection machine and X-ray inspection provides a non-invasive method of monitoring total GI residence time without affecting the normal GI motility. Mucoadhesive labeled with the technetium or indium or iodine have been used to study the transit of the drug delivery system in the GI tract. Similarly, we have another technique which comes by the name of gamma scintigraphy. It is a valuable tool in the development of a pharmaceutical dosage form, and with this methodology, it is possible to obtain information non-invasively. Finally, we have the bioavailability studies. Unfortunately, the bioavailability studies along with the bioadhesive system are not very encouraging. A few research groups have investigated the bioavailability of bioadhesive dosage forms using human volunteers or animal models. The results are a little controversial because some results were contrary to the in vitro results and some did not show the same extent of improvement as shown in the in vitro test due to the complications in the in vivo situations. Let us now try to summarize whatever we have learned in this module. It is but obvious that the concept of mucoadhesion involves the binding of a natural or a synthetic bioadhesive polymer to a biological substrate such as the mucous membrane. The success of any mucoadhesive drug delivery system depends upon the choice of suitable mucoadhesive polymer, their evaluation in proper manner also ensures their success, and hence usage of better evaluation parameters guarantees better success. If we have better evaluation mucoadhesive techniques, we are able to reflect upon the performance of the dosage form in a much clearer manner. There are various in vitro and in vivo methods which are available for better optimization of mucoadhesion. Combination of polymers can be tried and then sequentially evaluated for finding better mucoadhesion. Mucoadhesion allows the retention of the active drug over the mucosal surface and prolongs the contact between the polymer and the mucosal surface. Such properties could help to devise suitably tailored mucoadhesive drug delivery system and yield an ideal drug delivery because it not only will help in giving a controlled release but frequency of the drug administration is further reduced. Overall, it is going to definitely improve the patient compliance.